Sleepy Hope, Chapter 4. Fast Kill, Slow Kill. Dip the apple in the brew. Let the sleeping death seep through. Look! On the skin, the symbol of what lies within. Now turn red to tempt Snow White, to make her hunger for a bite. Disney Snow White, 1937. Known as the poison of kings and the king of poisons, arsenic developed its reputation as a discreet homicidal agent before the existence of the Roman Empire. Tasteless, odorless, and undistinguishable from food poisoning, arsenic was entirely undetectable by coroners prior to scientific advancements in the mid-1800s. No longer employable in the spheres of revenge and inheritance, arsenic took its skill set to a new territory, American farm soils, and locked in on its new target, the Colorado potato beetle. Early successes as an insecticide fostered increased demand for its services, and additional crop insect targets were added to the roster. Chemical companies branded and differentiated their arsenic products with cheery names such as London Purple, Shields Green, and Paris Green, reflecting their product's color. To increase potency, lead was often added to the mixture, along with sweet molasses and bran mash, to entice insects to eat it. Apply liberally, farmers were told, and they did. Sometimes by truck, but often with their bare hands. Unfortunately, insecticides aren't species-specific. They are dosage-specific, meaning their lethality isn't restricted to insects. Tiny insect bodies are more quickly overwhelmed by smaller quantities of insecticide than larger living creatures. Nonetheless, insects that do survive insecticide spraying have an advantage that we don't: their generational length and prolificacy. Generation can beget generation anywhere from a few days to a few months. What's more, they're highly prolific. Depending on the species, female insects can lay from a couple hundred to tens of thousands of eggs per day. Over a lifetime, some female insects will lay millions of eggs. Of these eggs, some will survive pesticide applications. Some will be just far enough away from the sprayer, perhaps resting on a neighboring fence post or in a culvert, to avoid a lethal insecticide dose. Other individual insects may survive through a slightly superior genetic ability to detox the insecticide from their systems. These surviving insects rapidly bequeath a swarm of progeny with now relatively concentrated superior detoxification genetics, and each successive surviving generation becomes better and better at flushing that particular insecticide out of their bodies. Thus, unlike humans, who have exponentially longer generation times and a relatively few offspring, insect species can quickly adapt to new insecticides. We call these genetic adaptations resistance. In the mid to late 19th century, as cultivated farmland increasingly emerged from former American forests and frontiers, arsenic applications to crops increased, and naturally, so did resistance. A vicious cycle ensued, and ever-increasing quantities of arsenic had to be applied to prevent crop decimation. While insects were building resistance to the increasing concentrations of arsenic, though, human beings were not. A tiny bit of arsenic won't kill you. Small amounts of natural forms of arsenic are even present in drinking water. Arsenic is dosage specific after all. Decades prior to the release of Disney's Snow White in 1937, American apple orchard arsenic concentrations were so high that Americans were reportedly literally keeling over from even a single bite of an American apple. By the 1920s, the U.S. government began quietly pulling arsenic-saturated apples from the market and then destroying them. As the horse and plow gave way to the tractor, and arsenic began to accumulate in American farm soils, came the 1930s Dust Bowl. Great Plains residents stuffed and restuffed blankets, sheets, and towels around window and door frames in attempts to block out encroaching dirt. The dirt got in anyways. Grasshopper hordes estimated at 23,000 per acre swarmed in and descended upon the scant remaining crops that humans and livestock alike looked to for survival. Mankind's survival seemed to hinge upon insect eradication. Despite the now well-known risks, we felt we had no choice and turned once more to applying more arsenic. How liberally did early 20th century farmers apply arsenic pesticides? Pesticide sales and applications have traditionally been both sporadically reported and underreported, and even are today. Records from the early 1900s through the 1940s indicate that at least 30 to 90 million pounds of arsenic-based pesticides were sold annually for application on American soils. Conservatively, around 1,200,000,000 pounds of arsenic blanketed the 400 acres of American cropland that spanned from New York to California over the first 4 decades of the 20th century. £3 per acre over 40 years may not sound significant, but arsenic doesn't break down in the soil. It doesn't wash away. It's persistent. It began to bioaccumulate. A new solution to the insect problem had to be found. In the fall of 1943, war refugees were hitchhiking their way to Naples, Italy, where U.S. and Allied troops had just fought and won a tenacious and costly battle to free the city from German powers. Once arrived, the refugees, along with Naples natives, sought safety from overhead bombing through underground living. Natives and refugees alike crammed into Naples' massive underground cave systems. The caves naturally lacked sanitation and were overcrowded. In the midst of squalid living conditions and with the people bordering on starvation, it quickly became apparent that on the heads of refugees had come another, less welcome guest: typhus-carrying lice. By December, nearly 400 Naples residents had contracted typhus. To protect U.S. troops that were guarding the city, the U.S. Army naturally wanted to quell the growing epidemic. The Rockefeller Foundation, along with a company called Monsanto, offered to help create a solution. They had a new powder that might be a miracle cure for lice. Monsanto's previous successes producing aspirin and food additives inspired confidence in their brand. Armed with this newly engineered lice-killing powder, the U.S. Army set up 40 massive walk-through spraying stations around Naples that were designed to dust up to 100,000 people a day. With the Rockefellers as chief financiers, by February of 1944, over 3.2 million people had been dusted with the chemical now known as DDT. Typhus and lice vanished. The success was unprecedented. The epidemic was declared over, and the operation ended. A little more than a year later, like synthetic inorganic nitrogen, Monsanto's DDT emerged from World War II in need of a job. Given its demonstrated success in Naples, its resume was quickly seized upon to eliminate the American agricultural insects that arsenic and lead had failed to conquer. If arsenic was applied liberally, DDT was applied lavishly. Frequently, DDT was mixed with diesel oil or kerosene and a 1 to 9 parts mixture. In one formulation, it was mixed with paint to apply onto farm buildings. Whether in the wilderness or on the farm, or within the home, it was regarded as not only effective, but harmless to children and adults. And like arsenic, farmers handled it with their bare hands. As air power became more accessible, we began spraying it down from crop dusters. The timing must have seemed a bit conspicuous. In the early 1950s, just as agronomists were trying to make sense of the massive Midwestern farm soil exodus, they appeared on the horizon. Like Hannibal and his troops, foreign, invasive, and previously unknown insect species rose up in mass to encircle, devour, and decimate crops. Worms, beetles, and moths, whether accidentally imported by cargo ships or planes across oceans or swept in by weather systems, began to hungrily devour crop roots, stems, leaves, and produce. No longer hindered by the natural predators of their native regions, these foreign worms, beetles, and moths began to hungrily devour food crops. Along with them came previously unknown strains of blight and mildew, wreaking similar havoc. Diseases of progress were how one chemical company labeled them. Cashing in on the Heartland's fears, Hollywood both amplified and made light of them by producing insect invasion flicks. Following King Kong and preceding Jurassic Park in true apocalyptic format, these films frequently paired a wise scientist with a love interest racing to stop these seemingly unstoppable insects before the inevitable decimation of crops and the Earth's human population. The beginning of the end, them, the deadly mantis, and insect fear, to name a few, left moviegoers with a petrifying, albeit illogical message. Kill one, and two take its place. Books and magazines spoke of the insidious efficiency of insects, relentlessly consuming our crops, infiltrating our homes, and undermining our health. The arthropods, one entomologist direly warned, would inherit the earth unless man abandons war and turns his martial interest to killing pests. Commentators regularly wrote of the war on insects, the invasion of pests, and the battles to exterminate unwanted species. Would it not be funny, one U.S. representative observed, to spend all these billions of dollars fighting communism and building the atomic bomb and then be eaten by the Argentine ant? William Setsu. The Green Revolution's phenomenal yields meant immediate security and prosperity, but only if they weren't wiped out by simultaneously increasing insect damage. The mantra remained that human survival and a rapidly expanding global population depended upon insect obliteration. The name DDT is somewhat of a misnomer. Its active, deadly ingredient is actually chlorine. The same is used in drinking water in swimming pools. That's probably why farmers felt comfortable amply applying it to their crops. How different was applying DDT to fields from municipalities applying it to drinking water? Chlorine sounded a lot like chloride, and in chemical terms, there's only one electron difference between them. The physical consequences of this single electron, though, are vast. Chloride occurs abundantly both in the natural world around us and is essential within our own human bodies, but chlorine is different. Chlorine is only found naturally in the deep recesses of the Earth's crust as a highly potent poisonous gas. By the chemical world standards, DDT isn't considered to be particularly toxic. Each and every DDT molecule contains only 5 chlorine atoms, hence the name dichloro-phenyl trichloroethane. Aspirin is considered to be more immediately toxic. The primary problem with DDT and other forms of chlorine, for that matter, is generally not your initial exposure. The problem was with your residual exposure. Pound for pound, the body is better at ridding itself of small doses of aspirin than chlorines found in pesticides like DDT and other PCBs or polychlorinated biphenyls. Chlorine is a part of a group of chemicals known as halogens that includes fluorine, bromine, and iodine. Because iodine and chloride are essential to both animals and some plants, multitudes of receptors for them exist within animal and plant tissues. Unfortunately, due to their structural similarities, both animal and plant bodies will mistake poisonous halogens for the essential ones, and once absorbed, they become extremely difficult to eliminate and may build up over time in an individual. It is from this family of chemicals that we often hear the term forever chemicals. In 1972, after almost 30 years, an estimated 1,350,000,000 pounds of DDT had been sprayed on a mere 900 million acres of American farm soils and foods. Public perception of DDT soured in the wake of Rachel Carson's book Silent Spring. Published in 1962, the book called public attention to the noticeable decline in songbirds that had fed on insects poisoned with DDT. Public reaction was so strong that the EPA banned DDT in the U.S. Largely in appeasement to public perception, the ban removed DDT specifically in other PCBs, but it didn't stop the U.S. and other countries from using halogen-based pesticides. Names changed, formulations changed, but pesticides actually grew more, not less potent. They had to. We weren't winning the war on insects. Just as with arsenic and lead, insects were developing resistance to halogen-based pesticides. With billions of pounds of arsenic, lead, and halogens having been poured out onto our American farmlands without much result, alternative methods became essential. Monsanto once again came to the rescue with a novel solution. Bacillus anthracis, the bacteria responsible for anthrax, and Bacillus cereus, a bacteria responsible for food poisoning, eye infections, and periodontal disease, are genetically incredibly alike. They have another close relation known as Bacillus thuringiensis, or BT. These genetic similarities between all three are so striking that some scientists have suggested that all three are actually the same species. Named after Thuringia, Germany, the location of its discovery, BT first caught scientists' attention when they observed its rapid lethal effect on its first observed consumer, a moth larva. Lightning struck. Why not replace bioaccumulating chemical pesticides with a bacteria to kill insects? What could be harmful to the environment about that? But how would a bacteria be sprayed on fields? It wouldn't. Bacillus thuringiensis would be bred into food. European farmers have a variety of names for it. In France, it's Brusson, meaning burl. In Italy, it's Rugna, meaning mange. German farmers cursed it with a virtual thesaurus of names, including Ausschlag for rash, Mauch, ugly, Krop for goiter, Raud for mange, and perhaps the most accurate, Krebs, meaning cancer. Officially, it's known as crown gall, and it appears as a cluster of regular plant cells grown wild into grotesque, cauliflowered, bulging lumps. Plants, like people, occasionally get tumors, big ones. A pathogenic bacteria causes them. At first, we thought this cancer-inducing bacteria only infected woody-stemmed plants. But scientists quickly discovered they could transfer this bacteria to a variety of softer herbaceous plants as well. Then we learned that these pathogenic bacteria have a truly remarkable ability to enter a host plant and transfer genetic material from the bacteria to the plant. This bacterium is what causes the tumors. The ability to transfer genetic information from one species to another was unknown prior to this. Animal bodies, in fact, are designed with a plethora of protections to resist invasions from foreign species genetics. Even when genetic transfer between similar species does happen, it doesn't progress to multiple generations. Wild herds of mules or packs of ligers don't exist, and with good reason. They are naturally sterile. Very rarely, the offspring of two different species, such as a mule, has produced offspring with another of the parent species, but that is where it stops. There are no further offspring. Scientists haven't failed in this pursuit for lack of trying. Even if scientists are able to create embryo offspring from a hybrid and a non hybrid species, and providing the female body does not reject the foreign DNA immediately, the female body of each species is so unique that the female will not be able to carry the offspring to term. Each mammalian species has a unique umbilical system or placental cotyledons. The number of cotyledons and the shape are unique to each species. Like electrical outlets and plugs on the same continent versus those on a different continent, the cotyledons of a mother and a baby from the same species match, but a baby and a mother from different species won't. Plants also have multiple defense systems to protect themselves and resist the transfer of genetic material from outside species, regardless of the origin. These environmental safeguards protect against environmental chaos and destruction. Changes to species DNA in nature are typically negative and harmful. That was true in this case also. The genetic change caused cancerous tumors on the plant. So, how did this incredible DNA transfer happen in spite of all these protective mechanisms? Some bacteria and occasionally plant cells were discovered to have a small disc, or mini genetic portal in their cells, known as a plasmid, which could receive genetic material like a book drop or a thumb drive. In nature, the genetic material these plasmids introduce would nearly always be harmful to the plant by creating cancer. Thus, the smaller the plasmid, the more protected the plant was. This particular bacterium has large plasmids, though. Scientists found that they could utilize these large plasmids to transfer the genetic material they wanted into the plant's cells. Perhaps they could have named it transbacterium to reflect the bacterium's primary function, transferring genetic material from one species to another. Instead, they named this bacterium agrobacterium, a bacterium that would transform all of agriculture. Agrobacterium was first put to work by transferring Bacillus thuringiensis into tomatoes. Tobacco, corn, potatoes, soybeans, cotton, and rice soon followed. When compared with continuing to saturate our soils with arsenic, lead, and halogen-based pesticides, Bt crops seemed like the lesser of two evils. Unarguably, in the early 1980s, genetics were conceived to be much simpler than we now know them to be some 40 years later. And even some 20 years post-introduction of commercial Bt crops, it was widely acknowledged that the genetics of Bt crop engineering were poorly understood. Though genetic engineers had access to this amazing genetic portal, they had little control in the insertion of these genes, meaning that inserted genes often flipped, were broken, or mutated in the process. Such insertions often inadvertently were observed to change the expression levels of natural genes. In MON 810 corn, for example, the promoter sequence that turns on the foreign gene was only partially incorporated into host DNA. The trans gene in MON 863 corn underwent some degree of mutation as well, and Syngenta's BT176 corn was supposed to create the Cry1AB form of BT, but analysis carried out both by French and Belgian government scientists showed that the transgene had only a 65% similarity to the intended protein. Jeffrey Smith, Genetic Roulette. Nevertheless, Bt crops were touted as a miracle to reduce dependence upon chemical insecticides. For a time, that was likely true. While they never replaced insecticides entirely, they did lessen our reliance upon them. But as with arsenic, DDT, and other halogen-based pesticides, Bt crops turned out to be just another 30 to 40 year stopgap. Bt resistance was always inevitable. Everyone knew that. This relentless chemical and genetic assault on insects has, albeit unintentionally, continuously and selectively bred insects with enhanced detoxification abilities. In the time it takes a pesticide company to engineer, test, approve, and market a new formulation, generations of insects have matured and reproduced young entirely immune to existing solutions. Some species have emerged with complete resistance to a new insecticide within as little as 2 years. At least 580 insect species are currently resistant to at least one insecticide. Some insecticide species have developed resistance to more than 90 different insecticides. Scientists can't keep up. Despite the miracle of Bt crops, the halogen family of chlorine and its beefier brother fluoride, and their cousin bromide, still dominate a broad array of widely used insecticidal agents today. Although insecticides are primarily classified by their mode of killing, scientists have even lately candidly admitted that they aren't completely certain how these chemicals work. Even after nearly a century of usage, the mechanisms of insecticides and their cocktails are still being theorized and debated. One thing is certain: we are losing this war. And we are losing it so badly that arsenic is being considered for reintroduction on American farm fields. We've come full circle. Famine by insect plague is no less an impending reality today than it was when we began pouring DDT on fields in the mid 20th century. If anything, famine by insect plague is more imminent today than it was nearly 200 years ago. Yet for the billions of pounds of arsenic, halogens, and Bt genetic material that has saturated our only 900 million-ish acres of American farmland over the past 100 plus years, insecticides actually comprise a small minority of pesticides used. Of the 700 or so agricultural pesticides currently utilized in American agriculture, the majority are herbicides. They kill weeds. That's the end of chapter 4. Thank you so much for listening. Please like and subscribe. That way you can be notified when the next chapter comes out. You can also find me on patreon.com. If you'd like to head on over to Patreon, that's where I'll be sharing more in-depth notes about each chapter and different interesting things I found during my research. Thanks again for listening.